Hello viewers. Today I will discuss about the protein structure with the aspects of protein folding, protein symmetry, subunit as well as interaction with respect to the protein dynamics. So I will talk about differently about the protein folding, protein structure significance, protein symmetry, aspects of protein structure, protein dynamics, protein subunit or protein assemblies as well as method to manipulate the protein structure as well. So we know that when the globular protein is folded, that obeys a geometric pattern and it is kinetically oriented. And we know that there are proper thermodynamics are there which predict the protein structure. Now in 1951, Lindström, who actually coined the term primary structure of the protein, have discussed the hydrophobic interaction is important for the protein structure to make. Next, in 1972, C.B. Anfinsen got Nobel Prize for his Anfinsen dogma, which actually tells us that the sequence of the amino acid predicts how the biological conformation of the protein will be made. I will discuss about the pattern of folding, kinetics of folding, dynamics of the folding, characteristics of the folding as well as various different other aspects of the protein folding. First, various information about the protein folding. The Anfinsen dogma, that is within the protein amino acid sequence, the message is carried that how the protein will be folded. So the experiment was that a particular bovine RNase was treated with 8 molar urea to destroy the hydrophobic interaction as well as beta mercaptoethanol which destroys the disulfide interaction. Now it was found that secondary, tertiary, quaternary, every structures are destroyed with this particular denaturing agent without the native conformation, that is amino acid sequence. Now when all those denaturing agents are removed by dialysis, the proteins will be automatically refolded back very easily, because how to fold this message was intact in the amino acid sequence. So that is the Anfinsen dogma. Now not only that the message carried by the amino acid sequence, but there are different propensities of the amino acid sequence to fold either in alpha helix, beta sheets, beta strand or beta turn as well. And there is no half folded protein during the course of protein folding. There is the equilibrium in between the folded and unfolded structure of the protein. Next, pattern of folding. So you can observe how the different types of the helices, beta sheets, they are joined by the large polypeptide to make the local loop. It may be single domain or multi-domain. The multi-domain are found in case of made by solely alpha domain like tobacco mosaic virus coat protein, or beta domain solely made by the beta strands in case of pre-albumin structure or parvalbumin as well, and mixed alpha and beta structure is also found in case of various thioredoxin, glutathione reductase as well as different types of protein. Next, formulation of general rules how the proteins are getting folded. The general rule is that the hydrophobic residues like isoleucine, valine, they will be kept inside. They will be buried inside to make hydrophobic interaction. And over the surface, hydrophilic residues are there like aspartic acid as well as lysine, etc. and serine threonine to make hydrogen bond. Very important is this that beta strands actually are twisted. In case of silk fibrin, we can follow this case of twist in barrel and protein structure. The open beta strands are found at the N and C terminal end, and it actually helps to change, abrupt change in the direction of the protein. In case of beta corner also, which actually also help to abrupt change in the protein direction, because one strand may contain glycine, other strand may contain beta bulge as well. We can find the presence of the prosthetic group that actually enhances the folding, because in case of hemoglobin, myoglobin, the prosthetic group heme is actually present the twisted structure. Next, thermodynamics of the protein folding. It includes the conformational entropy, conformational enthalpy, as well as interaction of the hydrophobic residues, hydrogen bonding, van der Waals interaction, disulfide interaction, ionic or charge interaction, because you know in case of subunit interaction, this salt bridge or ionic type of dipole-dipole interaction is very important. Now, characteristics of protein folding. From this graph, it is known that the protein folding is cooperative in nature. Okay, so a certain jump from folding to unfolding can be followed. Leventhal paradox is actually difference between the calculated and actual folding, as well as incorrect intermediate during the course of the proper protein folding. Next, pathway of protein folding. Either this particular protein can be folded in the two-state model or multi-stage model from the folded to unfolded structure. In case of two-state model, we can follow there is a sharp transition within single relaxation time and temperature dependent process. And in case of multi-stage model, we can follow there are different types of interactions are there. In case of multi-stage model, the quaternary structure is monitored by light scattering and the polypeptide chain formation is monitored by CD spectra. In case of glutamate dehydrogenase, from hexamer to trimer, the process is reversible for the unfoldedness. But from the trimer to monomer in the native conformation, it is very much irreversible. Next, antigenic detection. And that is very important during the course of refolding. Sometimes some presence of the metal ion is needed. Zinc is needed for the carbonic anhydrous reaction, which actually speeds up the proper folding. Antigenic detection is actually tells about there is equilibrium in between the folded and unfolded structure. Now, mechanism of protein folding. As nucleation event occur, so the transitory nucleation mechanism can be followed, and it is very much kinetically controlled pathway and pathway dependent and thermodynamically dependent pathway. It is very important that after protein folding, some covalent modification, cleavage of peptide bond, etc. are very important. So, during the course of refolding, that also be taken account. Dynamics of protein folding. Here, the kinetics of protein folding is included, importance of the disulfide bond as well as chaperone action is very important. Kinetics of protein folding. As it is a pathway dependent kinetically important process, a sharp transition can be followed during protein folding and unfolding process. And the intermediates can be trapped as molten globule, nucleation as well as final packed structure. We can follow the presence of various types of undulation, because globular protein is not static, the internal molecular motion is there. And another very important thing is that it is very rapid process, but sometimes it is delayed by off-path processes. That off-path process includes the cis-trans isomerization of amide bond of the neighboring proline residue, as well as the incorrect disulfide bond linkage intermediate can also delay the protein refolding. So, disulfide bond, it is very important for the proper folding of the protein structure, either tertiary or quaternary or protein folding. The importance of the chaperone action during protein folding. So, what is chaperone? They are nothing but the guide protein. They actually do not carry the protein folding, but they guide, they protect the protein folding, because how to fold, that message is given inside the protein structure. The protein automatically can be folded, but within the chaperone structure, they are actually folded in the protective environment. GRO-EL and GRO-ES complex is very important for the protein folding. GRO-EL make the hydrophobic core, right? The ATP hydrolysis is needed. There is the presence of 2 rings having 7 protein structure for the GRO-EL, and it is capped by the GRO-ES. So, Anfinsen cage for the protein folding is maintained in vivo condition with the help of ATP hydrolysis. So, protein-protein interaction is very important. It is very tight binding among the different subunits, and 20 FM dissociation constant is for the ribonuclease and its inhibitor binding. We can follow various different type of interaction like homotypic and heterotypic interaction. In case of homotypic interaction, very distantly situated subunits are getting interacted because they are somehow asymmetric, and nearly symmetric interactions are found in heterotypic interactions. Next, protein families. The protein structure is very much needed for a particular sequence to give the protein structure. So, these are all structures are included within the protein family. Now, it can be known that they can be diverged from the common mother protein, either according to the sequence and structural similarity. But sometimes for further folding, some protein structure can be also converged. So, these sort of protein families, protein structure, three-dimensional structure prediction of the protein family is evolutionary related. Next, protein structure function relationship. As we know that proper folding is very much important for a particular protein biological action that is found in calcium binding site of the calmodulin. We can follow the sequence structure relationship in three-dimensional space. As we know that Roseman fold, there beta alpha beta motif, there are different types of folds are there responsible for different supersecondary protein structure and function. They are kept in protein database. So, they can be further utilized to know how the functions can be known from the structural database analysis. Implication of the protein structure. So, there is a molecular evolution, DHFR, dihydrofolate reductase enzyme, very much important and present in both prokaryote and eukaryote. So, this sort of implication of protein structure can be done. Number 2, very important unstructured protein that is present intrinsically. So, intrinsically unstructured protein can show its property when it is interacting with other protein structure. And biologically unstructured protein is very important to show the prion action in mad cow disease. Usually PRPC is found, but when it is converted to scrapey structure, PRPSC, here you can follow there is change in the assembly at N-terminus end. Some beta turns are made, but some are not. So, in this way, the biological implication of this protein structure is very much important. As we know that this particular thing is very much controlled via several environmental factors like pH, like voltage, etc. So, that is the environment control conformation. So, we must take care of because all the proteins biological action for their structure is related to chromatin modeling as well as transcription and other aspects. So, here we can find rotational as well as helical symmetry. Rotational symmetry, cyclic symmetry, dihedral symmetry, octahedral, tetrahedral, all those symmetries can be found. As well as if the symmetric interactions are there, it indicates type as isologous symmetry, or heterologous symmetry, that is non-symmetric, unsymmetric interactions are actually followed. Now, in all the cases of symmetric reaction, what is point symmetry? Where all the subunits have same geometric type of symmetry, same type of conformation. So, that is called as point symmetry. And these twofold symmetry is found in case of hemoglobin, where two units, two alpha, two dimers are getting interacted together. Now, aspects of protein structure. It includes the allosteric regulation, receptor theory as well as enzyme activity as well. First, let's talk about the allosteric regulation. Now, what is allosteric modulation or regulation? Suppose in multimeric protein, suppose the conformational changes happen in one subunit, so that can be manifested to the same conformational changes to other subunit. The structure function all changes together. Actually, there are two types of conformations are there, rotamer or having different input of energy. What are they? Tensed T state as well as relaxed R state. Now, binding of ligand or substrate actually favors either T state or R state. Now, let's talk about the model for allosteric regulation. What are they? Number 1, concerted model. Number 2, sequential model, as well as there is number 3, morphine model also. What are they? In case of concerted model, that is actually also termed as monode model or symmetry model. That is the change in one particular subunit conformation changes to the conformational changes to other subunits also. So the subunit interaction is very important. That is the symmetry model. Actually found in case of hemoglobin. Oxygen binding to one subunit changes, that is the cooperativity nature, changes the interaction to other subunit. That is also called as allostericity also. Allo means at different sites. So it increases the oxygen propensity to other subunit with high oxygen pressure and release when the oxygen pressure is low. These particular thing we can follow, there is equilibrium in between T and R state, and the binding of ligand or oxygen favors the relaxed state. The next model, very important, sequential model or Coastland model. It does not obey the thing that is change in conformation of one subunit changes conformation to other, because all conformer subunits are not identical having same symmetry. That is actually applicable only for monod model, concerted model. It actually, sequential model obeys the induced fit kinetics, induced fit model of the enzyme catalysis. The third one, morphine model, the homo-oligomeric interaction it obeys, and dissociation of concerted model. Next one, allosteric regulation, either homotropic or heterotropic. Homotropic means itself substrate or ligand acts as effector. Suppose oxygen in hemoglobin, this is the substrate or ligand, but it also increases the affinity for other subunit to bind with oxygen. So it is homotropic regulator, allosteric regulation. Heterotropic means other than the substrate or ligand, which can change, which can affect the regulatory side binding towards the allo side binding. Now here what are the examples? Like H plus, proton, carbon dioxide, 2,3-BPG, they are the heterotropic regulator. Binding to them, to one subunit, that decreases the affinity of oxygen to other subunit. Next, allosteric modulation may be positive modulation that increases the allosteric affinity. Suppose ligand affinity, like in case of oxygen binding to one hemoglobin subunit, increases the affinity towards the other subunit. Or negative modulation, that is allosteric inhibition, that is done by 2,3-BPG towards the oxygen binding in hemoglobin. Now these allosteric modulation is very important for allosteric resources. They make the database to know protein structure and function, as well as they are very much important for drug target site like GABA neurotransmitter to G protein couple receptor, as well as they have different other functions. They are very much important for pharmacology also. Non-regulatory allostery. Suppose sodium is needed for the protein, between different types of protein structure formation. But the problem is that how it regulates by inputting and removing, that is unknown. That is called non-regulatory allostery. Receptor theory. Postulate and different model. Postulate is what? The receptors are very structurally and sequentially specific towards the ligand binding. Number one. Number two, high affinity towards ligand binding. After binding to ligand, the changes in structure, biological conformation, biochemical changes happen within the receptor, and it maintains a saturable kinetics. The limit is there. There are two models are there, like two-state model as well as ternary complex formation model is also present. In case of two-state model, what we can find, not only ligand or receptor but also activated receptor R dot is also present. Next, enzyme activity. As we know that enzyme is very specific at the active site to bind with a specific substrate. In some enzyme, regulatory site is there which controls the activity of the active site in the catalytic domain, catalytic site. Here we can follow the enzyme substrate transiently, reversibly changes its shape, structure for the speeding up of the reaction. But after reaction completion, it also by the product release gains the own structure in case of enzyme. And enzyme regulation can also be demonstrated by cooperativity as well as allostericity, function of subunit. Protein dynamics. The protein, globular protein is not static, okay. So some energy input, time input is needed for the protein to be properly folded. So the largely distant molecule will come closer. That is actually helpful to describe protein activity, regulatory as well as catalytic activity together. So these things can be presented by Anfinsen experiment as well. characteristics. So it includes the local flexibilities of the residues. So the chemical modulation is there, chemical undulation, chemical bonding is present, as well as regional flexibility, intradomain multi-residue coupling attached via hydrophobic interaction to make the hydrophobic core. The global flexibility, which is imparted as the flexibility or the mobility of the multi-domain complexes as well. So different proteins have different types of this sort of protein assemblies together. In case of protein subunit, we can follow, that is the co-assembly of different subunits together. That may be the small association oligomer, it is called as in case of hemoglobin, or multi-subunit protein like quaternary structure, it is followed as microtubule formation. So they can be swapped by the genetic engineering, can be separable as they are co-assemblies, and it follows one gene, one subunit hypothesis. They may be single domain or multi-domain in case of protein quaternary structure. This is the picture of ATP synthase. Because the domain is nomenclatured according to the Greek letter alpha beta, it is hexameric alpha 3 beta 3 as well as C ring structure. ATP synthase F0 F1 ATPase needed to generate the ATP. Next, subunit interaction and determination of the composition. The subunit interaction can be monitored via cross-linking with glutaraldehyde, because the subunits are buried 100 to 200 angstrom within the core and they are interacting same non-covalent interactions. Though they are separable, they can be monitored very well. And the subunit composition is very much well determined via protomer oligomeric structure that is made within the protein database data bank, from where the structure function analysis can be very well coordinated. And subunit composition can be known via various different types of methods, like light scattering as there, like cusp determination are there, the computer modulation, different techniques they involve to know the subunit composition. The attempt to manipulate the protein assemblies. The protein assemblies can be manipulated by changing pH of the environment, by changing solvation power, the ability of the protein to interact with the solvent as well, and it is transient metal dependent. Sometimes transient metal can manipulate the protein structure formation. The subunit interaction of the protein can be monitored via X-ray crystallography as well as NMR study. So students, I have discussed about various aspects of the protein structure with respect to the protein folding, make protein assemblies or subunit, and how the protein dynamics it is related. Thank you very much.